Forest to bridge.
This is the Enterprise.
Captain, I'm switching on the transmitter.
Extraordinary. Tony, what's that? Over to the left.
What?
Yes, that. No, back. Yes, that, right there. What is that?
That's Commander Riker.
So that is a visual prosthetic. Sorry, representative in the fictional depiction in Star Trek: The Next Generation that's supposed to take place in the 24th century. And as you can see from my title of this talk today, Brain Stimulation for Restoration of Vision to the Blind, The Next Generation. That's what I want to talk about today, is a little bit of the work we've done, and excuse the take on Hollywood, about this very challenging problem of introducing visual percepts to the brain to restore vision from the blind. And as Daniel pointed out, it is a very challenging task at any level of input, whether it's at the retinal level or at the visual cortical level. And I want to talk about our experience in this work over the last decade or so. So I'm currently at Baylor, as Daniel said, but I will be moving on to the University of Pennsylvania in just a few weeks. But all the work that I'll show today was done either at Baylor or with my collaborators in California. I've received some funding from the National Institute of Health, DARPA, and the VA, and I do have, in terms of conflicts of interest, a relationship with Second Sight Medical Products, where I've been a PI for their clinical trial of the Orion, the visual cortical prosthetic that this company has developed, but I have no financial interest in the company itself. So I think everyone in this audience is well aware that visual loss is a daunting and challenging problem in society that affects thousands of people across the world. And in most cases of acquired blindness, the visual machinery itself, the eye itself, is damaged and damaged irreparably, but the visual cortex, with most of the processing, or much of the processing, I shouldn't say most, because of course a lot of processing occurs at the retinal level, but the brain itself is intact and functional. And for decades, really, it's been a tantalizing proposition to the scientific community, the idea of using a visual prosthetic to introduce information directly to the brain and help patients have some restoration of visual function. And while retina is a wonderfully tantalizing target in itself, in many patients, the retina is damaged in many blind patients, and the visual cortex in some ways is a more desirable target for a prosthetic device because there's more real estate available for implantation of electrodes thanks to magnification factor. So we've known that an effective VCP, or visual cortical prosthetic, is desirable, but why do we think it's possible? First of all, electrical stimulation of the visual cortex produces a small, bright spot of light, a percept of a small, bright spot of light known as a phosphene. And this slide here shows the distribution of all the points on cortex that Canadian neurosurgeon Wilder Penfield found produced visual sensations when stimulated in patients in the 1930s and 1940s. And these sites, as you can see, are distributed over a rather large region of the occipital cortex with the highest density. Can you see my arrow as I point?
Yes, yes.
Okay, with the highest density towards the occipital pole. And even back in the 1950s, it was recognized that electrical stimulation of visual cortex could potentially be exploited to restore vision to the blind through the development of a prosthetic device that would target visual cortex. So that's one reason that we believe visual cortical prosthetics have potential. Stimulating the brain produces phosphenes, or visual percepts. The second reason is that early visual cortex contains precise maps of the visual world. And you can see these maps right in front of us. For example, if we extracted and flattened V1, shown in red in this figure, from its location in the medial occipital wall, we can examine how visual space is mapped in V1. The upper visual field is represented in lower or ventral V1, the part beneath the calcarine fissure, and stimulation of the sites in yellow and red produces phosphines in the upper right visual field. The lower visual field is represented above the calcarine fissure, and stimulation of these sites, shown here in blue and green, lead to percepts of phosphines in the lower right visual field. So this is really the basis for the theoretical attraction of developing a visual prosthetic. The ability to produce phosphines plus the existence of a precise retinotopic map is the basis for this development. And the idea is if we stimulate one electrode in one location within the primary visual cortex, here marked in red, we get one phosphine in a predictable location in the visual map. So the hope is that if we stimulate a large number of electrodes located over visual cortex, we might produce a rudimentary form of a vision, here idealized as an image of Jonas Salk. So what would a visual cortical prosthetic device look like? The main components would be some sort of camera on a pair of glasses that the subject would wear, pre-processing circuitry, somewhere to convert raw signals into those that will be inputted into visual cortex, and then transmission of the data to electrodes implanted in visual cortical areas, which would deliver a pattern of stimulation designed to produce a useful visual percept. So this has been tried before, I mean, with some really rather impressive efforts going back to the 1960s. And as you can see in this figure, technology was a major limitation. And this is one of Brindley and Lewin's subjects. And obviously with all this equipment, the subject could only be tested in the lab. It was a pretty remarkable accomplishment in the 1960s. All the actual data transmission was wireless, although the subject had to wear this large helmet with transmitters to send wireless signals. Quite impressive in some ways, but certainly very restrictive in others. The next group that worked on this quite famously was the group led by Dobell. And in this device developed by Dobell, the subject could now wear a portable computer and glasses and could move around in his environment. And despite the limited technology of these times, these devices did have some success. Subjects could perceive phosphines corresponding to the stimulation of individual electrodes, and they could learn to accomplish some tasks using the device, though the documentation in that era was not as rigorous as we might have liked. A third effort, several decades ago still, was performed at the NIH and used penetrating electrodes implanted in a single subject for a period of months. Penetrating electrodes did allow for lower stimulation thresholds and smaller phosphines, but techniques to develop penetrating electrodes that are safe and reliable for the long term are still very much in development, and this did not end well for this particular subject. The device failed to have consistent functionality just after several weeks. So what's going on right now in the field? Well, because of the dramatic increases and advances in technology, there's a lot more interest in this field just in the last decade or so. And there are at least 5 groups that are working on projects designed to develop a visual cortical prosthetic to be implanted in human blind subjects. And this is not even touching the impressive amount of work going on that targets the retina. There's of course a group in Australia, the Monash group, a group in Chicago at the Illinois Institute of Technology, the group led by Peter Rolfsema in the Netherlands. And then there are 2 groups that we've been actively working with. One is the group at Columbia led by Ken Shepard and funded by DARPA, where we're involved in the clinical testing as the work moves in that direction. And the other, the one I'll focus on quite a bit today, is the impressive work done by our colleagues at Second Sight, based just outside of Los Angeles, California. And this work is funded by the National Institutes of Health. And there's an early feasibility trial underway as we speak, both at Baylor College of Medicine and at the University of California, Los Angeles. And some of the data, a small amount of the data, one subject in our Cell paper was work done in one of these subjects implanted with the Orion device, and I'll show you that later on in our talk. So what are the problems with the existing visual cortical prosthetic devices that we have? What's been holding us back from achieving real form vision, which is of course the goal? What are the limitations in the early devices and to this point in the current devices? So we can talk about two categories of problems. They're the hardware problems or technology issues, the number of electrodes. For example, here in the top left, you can see what the Orion device looks like, the part that interfaces with the brain, and there are only 60 electrodes. There are other problems in the past related, for example, with the Utah array imaged over here with the biocompatibility and longevity of electrodes. This is especially a problem with penetrating electrodes. Another category of problems, of course, is software related, and that's the way we like to look at it, hardware and software. Or when I say software, I mean how we use the device to input information to the brain. And the prevailing assumption over many decades has been that you take an image from a camera and essentially dump that image on the brain as if electrode stimulation was identical to pixel activation on a video screen. And we have found, and I think Daniel alluded to this in his introduction, that it doesn't work that easily, that for a variety of complicating factors, it's not easy to simply treat phosphenes as pixels. So we believe that both the hardware issues and the software issues must be addressed to produce a modern visual cortical prosthetic that really will have some value in changing subjects' life. We have to improve the hardware side and the software side. In my talk today, I'll focus mostly on how we've approached stimulating the brain, the software side, and I'll show you some of the research we do both in two types of subjects. One is sighted patients with medically intractable epilepsy who are just in an epilepsy monitoring unit for clinical reasons and who allow us to use this opportunity to test stimulating paradigms on the brain. And the other category are blind subjects, largely taken from the Orion clinical trial that is geared, and both these two subjects offer valuable approaches to tackling these questions related to the development of useful stimulation paradigms. So let's look at electrode resolution from the perspective of what we can produce with varying number of electrodes. So this image here shows a 5x5 array of imagined electrical stimulation of the brain, 25 electrodes or 25 pixels. Now I'm going to walk you through a sequence of images. So here you see 64, 8x8. And this array would correspond roughly to the number of electrodes in the only FDA-approved device for clinical testing that exists. And that is, of course, the Orion visual cortical prosthetic. And as you can see, or can anyone see a structure evident in this image? I think it's quite difficult. And this is what we have. This is what we're testing with right now. Let's jump ahead. This is a 10x10 array, equivalent to the Utah microarray. Here we jump ahead to a 25x25 array. And some groups plan on using multiple Utah arrays or similar systems in their proposed systems. Anyone recognize what we're looking at yet? Here we get to 1600 pixels or 40x40, 2500. Wow. And here we get to 100x100 or 10,000 electrodes or pixels. And this is 10,000 electrodes more than imagined by any other system, any existing system. And we'll jump ahead maybe into the realm of science fiction. Here we have 200x200. Here we have 500x500. And here we have President Mandela in his full glory at 1,000x1,000. This is 1,000,000 pixels, analogous perhaps in a crude sense to 1,000,000 electrodes. And that is what we have planned for a device that we're working in collaboration with Ken Shepard and his group at Columbia to develop. It's important to note, we don't expect real visual cortical prosthetic systems to be able to fully present images like this for many reasons. First of all, there are gaps in the visual field due to folding of visual cortex and blood vessels. There's variability in the size, brightness, and texture of individual phosphenes. interactions and what happens in the visual cortex when multiple sites are stimulated. And we're inputting information to the visual cortex in a much different way than what occurs in natural vision with inputs from the normal retina to cortex route. So in short, more electrodes is probably a critical step in the development of useful visual prosthetics, but it in itself is not enough. We are inputting information to the visual brain, and we need to optimize how this information is sent through electrodes to optimize perception. So the next couple of slides give you perhaps a sense of the limitations of standard methods of stimulating visual cortex, which we would summarize as simultaneously stimulating the visual brain with many electrodes at the same time. Our approach is to use a new technology down the road with thousands of electrodes implanted on a flexible sheath, and to re-envision or reinvent how we deliver that electrical stimulation. But before we get to the future, I want to go back a bit to the past and show you what we've done to test some of these concepts. So this is a slide that kind of shows you a little bit about the research we've done in the past using subdural electrodes in human epilepsy patients. So you can see here a large craniotomy, and at the margin of the craniotomy, here is an electrode tail. And under this margin, going around the back of the visual cortex, is this hybrid designed electrode that includes clinical electrodes that are 3 millimeters in diameter, along with what we call our research arrays that are, we use the unused space in between the normal clinical electrodes to implant a small grid of research mini electrodes that are 0.5 millimeters in diameter. And this allows us to test some of the paradigms that we were working on. And this technique is useful using these little arrays because it has high spatial and temporal resolution. And while they're sitting in the EMU, the subjects can perform multiple behavioral tasks with little training, much easier than working with monkeys. The subjects can actually report to us what they see, which is so valuable in this kind of work. And we can record from lots of brain areas all at once, including areas outside of visual cortex. As you see, there is a lot of sampling. In this particular case, this is the temporal lobe, and here's the frontal lobe. And we can record activity we can not only record activity to correlate activity with perception and behavior, but also use electrical stimulation to actually alter activity and behavior. And in the EMU, we do both recording of data while subjects view certain visual stimuli, for example, to match visual receptive fields. Sorry about that. And we also do electrical stimulation. So here we're presenting a visual stimulus and mapping the, presenting a visual stimulus and mapping the site, the receptive fields as represented in the visual brain. And here we're actually stimulating the brain and having the subjects report by drawing on a touch screen the dimensions of the phosphene that they perceive in that particular spatial location. So which areas of visual cortex produce phosphenes? You have to remember that earlier I showed that slide from Penfield that showed the region of the cortex that he found that produced visual percepts. But of course, in Penfield's day, very little was known about the organization of visual cortex. We now know that the visual brain has a complex functional organization and contains multiple visual areas shown on these images. And in the past, we used a very precise approach to define which visual areas actually produce percepts. And what we found is that early visual areas, including V1, V2, and V3, consistently and really reliably produced phosphenes at relatively low currents. And as we got to later visual areas, it became increasingly difficult and in fact quite rare to produce a visual percept without training. And this is of course relevant to the eventual development of the visual prosthetic. Let me jump ahead a little bit. We were able to develop mathematical models quite simply that allowed us to predict phosphene size and location based on measurements of the amount of cortex activated as well as the magnification factor. And we wanted to jump ahead at this point and test the hypothesis that we could just use the classic concurrent stimulation of multiple electrodes to produce a useful visual percept. And this is sort of a figure that's really lifted from an earlier NIH grant of ours, where we said we would take electrodes in epilepsy subjects and stimulate concurrently multiple electrode sites at the same time, after defining, using spatial mapping with both receptive field maps and phosphene maps, the actual locations of where the phosphines would be produced with stimulation of individual electrodes one at a time, and to see if we can match the sample and produce useful percepts. And when we tried this approach, as Daniel would have readily predicted, it worked rather poorly. Now, there's a cautionary tale historically in this, and Brian, of course, remembers, and Brian and I have talked about this work many times. The best Dobell really was able to do and prove with rigor, and I'm not talking about Dobell showing subjects driving, blind subjects driving cars in empty parking lots, but what he showed with rigor was this paper in Science, where he taught the subjects with extensive training to recognize patterns of phosphines in association with 26 letters in the alphabet. And he called this cortical braille. And why do I call this a cautionary tale? Because Dobell did not ever report that he was able to stimulate multiple electrodes at the same time and produce an actual percept of a letter. He was able to train the subjects to learn a code representing letters. They could represent a collection of disparate dots, as shown in this figure. When we tried simultaneously stimulating multiple electrodes at the same time, it was very difficult for the subjects, and these are sighted subjects in the EMU, to draw them all at once. And we would have to stimulate all, in this case, all 5 electrodes at the same time. And over multiple trials, the subject would draw the spots, but they never were able, with all our efforts, to produce a coherent percept of a desired letter. Now, some of this, of course, could be related to electrode spacing and the crudeness of what we were working with. Some of it could be related to attentional factors. The subjects were not seemingly able to attend to all the electrodes, the phosphines, at the same time. There are a wide range of factors that could have produced this limitation, but we worked on this for a long time and really became frustrated and disappointed with our results of simultaneous stimulation of multiple electrodes at the same time. So then we asked the question.
Daniel?
Yes, I'm sorry.
Hi, this is Kalanit. Good to see you.
Hey, Kalanit, how are you?
Hi, I'm sorry to interrupt here, but I have a question that's been on my mind.
Sure.
So, how do you deal with this log polar transformation between the world and V1? Because you didn't mention it, and I'm sure you've thought about it.
I'm sorry, what transformation?
Log polar, right? So it's in polar coordinates on the cortex, and there's foveal magnification.
Right.
So I just was curious about how you guys deal with that.
Well, you can see that in this particular case, look at the differences in the sizes of the phosphines. It's a problem. We don't have, an idealized version of a visual cortical prosthetic would have different electrode sizes corresponding to the variability of magnification factor across visual cortex. This is just a crude attempt to deliver a very basic coherent percept. We were not dealing with that level of robustness of a visual percept.
Okay, got it, thank you.
So what the idealized pixelated letter we wanted to produce was something like this. When we stimulated electrodes that corresponded roughly to this distribution across visual cortex, this was really what we typically achieved. And what we really wanted to get is something more in the order of this. Now, what we decided to do is take a different approach. And then this was motivated by 2 problems. One is that we have gaps between our electrodes positioned on visual cortex that might have contributed to our difficulties in producing a coherent percept. And another problem we have is that even when we do produce tightly physically or spatially located phosphines, it's difficult to get them to be perceptually coherent in our experience. So we developed a technique based on the classic medical school teaching. When you want to test someone's somatosensory function, one of the simplest tests we do is we have a subject, we draw a letter in the subject's hand, and if you try to probe a patient's hand using the somatosensory equivalent of pixels or focal presentations on the hand, they don't perceive a coherent percept of a form. But if you trace it along their hand, they do it very easily. And we thought that this might be a viable approach to both stimulate rapidly and sequentially, and also to use current steering to create virtual electrodes in between the sites of the physical electrodes that we implanted. And this was an approach we took as an attempt to create coherent percepts versus a few isolated blobs that could never seem to produce anything in the way of a desired visual form. So here's an example of the individual maps of phosphines from various electrodes in a particular subject. And I'll play you this video of how we stimulated the So our attempt was to stimulate these electrodes in sequence according to this map to produce a letter. And as you can see, and this was the first time we had this success, it was very gratifying when the patient drew exactly the form we intended, corresponding to this pattern of stimulation that I've just traced along the visual, the phosphine map. And the patient readily was able to say that looked like a letter Z to me. And we proceeded to do this in other subjects. Here's another epilepsy subject. In this case, the subject was implanted with a much finer array of 24 mini electrodes on the medial wall of the occipital lobe, as you see here on the top left. And using the electrodes in this array, we designed 4 dynamic stimulation patterns corresponding to 4 different letter forms. And these subjects notably had no real training at all. And without any training, the subjects were able to perceive these patterns of dynamic stimulation and reproduce them on a touch screen. And here you see examples of each of these desired images. And the patients had a striking correspondence between the predicted and actual letter percepts. And we did some very basic psychophysical testing of the subject's ability to discriminate between these patterns using a 4 alternative force choice task. And this particular subject was able to perform the task with 66% correctness, significantly better than, of course, 25% chance. And that was just working with this subject over about 2 hours with no training and just really, it was rather intuitive to the patient, which was quite striking. It seemed really quite accessible. So moving on from sighted subjects in the EMU, our collaboration with Second Sight gave us another potential avenue to test this. So this is a young woman who has acquired blindness. She was not implanted with an Orion device. This was a step before the Orion was implanted. This young woman was implanted with a NeuroPace device, which is a 8 electrode device with large electrodes, 2.5 millimeter diameter electrodes that was actually developed clinically for epilepsy. And this patient volunteered in working with my collaborator, Nader Paratian at UCLA, to undergo some testing of electrical stimulation in visual cortex in a blind subject as a precursor to implantation of the Orion device. So this is an image of the phosphine maps. And for example, you can see electrodes 1, 2, and 4. The map is below the horizontal meridian, while for 3 and 5, it is above the horizontal meridian, as one would expect. And we use these very crudely sized and spaced electrodes to see if we could get any results with dynamic stimulation to approximate percepts of a visual grapheme, a desired grapheme. So let me show you an example of what we were able to do with her. This is a dynamic pattern using 5 electrodes, and we asked her to draw what she was seeing. And here we tried to do an upside down U. And you can see, notably, and how rapidly and confidently she draws the pattern after a trial of dynamic stimulation. So this was very encouraging for us. And this was a wonderful subject. And since we only had 5 electrodes in this particular subject that produced phosphines, we developed an array of stimulation patterns for these 5 electrodes to try to produce different percepts. And you can see, and we kind of worked with her to give them names that she could identify for testing. And you can see each different color here is an individual trial. And we did use the dynamic stimulation of those 5 electrodes in different orders and different sequences to produce 7 different patterns, 6 of which are shown here. And to assess reliability, the patient was stimulated repeatedly with each of the 7 stimulation patterns randomly interleaved, drawing the perceived pattern following each trial. And different shades of blue show different trials of the same pattern. And when we had her do a forced choice discrimination task with 5 of these patterns, she obtained the 5 that were most easily recognizable to her and translated most readily to her as a letter. She was able to perform this forced choice discrimination task and get 93 of the stims correct, while chance, of course, would predict just 20% correct. And again, this was with very limited training. We had more of an opportunity to work with this patient because this was a implanted device. It wasn't a limited window of a few hours of time in an epilepsy monitoring unit. But it is striking that we were able to achieve this with just a couple of hours of training to get this level of performance. And this was just dramatically different from what we had achieved in the past when we tried to stimulate multiple electrodes at the same time to produce a percept of a visual form. So,
hey, Daniel, can I ask a question?
Sure, please.
This is Bill Newsom. Hi. Really cool result there. I wanted to ask about the timing of the stimulation from electrode to electrode.
So on that particular case, the timing was, and I actually don't have the numbers in front of me, the timing was quite slow. And the reason for that is the limitations of the software. We were just using a software designed to stimulate patients to abort seizures. In later subjects, and I'll show you that data shortly, when we worked with the Orion device, we were able to stimulate at a much faster sequence and still get very ready discrimination of forms. And obviously, we're limited by the devices and the exact temporal speed with which we can stimulate. And that one, I think, was on the order of less than 10 per second. It was quite slow. We become faster later on.
So her fingers, the upside down U she was drawing, were more or less done in real time as you were moving this?
This is more or less in real time. Yes, that for sure. This is the slowest, and she's drawing in more or less in real time.
Okay,
yes.
Thank you.
And I should point out, Bill, you see how she has her finger on this little, that's what I call in Texas a nubbin. What we did is we wanted to eliminate or minimize problems that Avi's very familiar with, problems with eye movements. So we would have the subject fixate, quote unquote, fixate on a palpable spot on the screen. So she would touch that spot and fixate and then draw from that point. And that seemed to work fairly effectively. Obviously, this is an issue that's going to be a major concern in the future. Eye movements are a very serious potential confound. So the Orion device, as I talked about earlier, we're a part of a feasibility study for this device that's made by Second Sight Medical. This is the same company that previously made the Argus 2 retinal prosthetic. And there are 2 testing sites for this device under an FDA-approved clinical trial of the early feasibility of the device that's ongoing, one at Baylor and one at UCLA. And these schematics show the organization of the visual cortical prosthetic, 60 electrodes, as I told you before, and they are located in the subdural space on the cortical surface. The tail of the array goes out to electronics and a receiver coil located on the lateral aspect of the skull over here. And then the subject wears sunglasses with a camera and a visual processing unit that communicates to a transmitter that is placed over the implanted coil, which you see over here. And real-time video captured by the camera is downsampled and processed by the VPU and then converted into stimulation patterns based on the brightness in the scene, and the data and power are sent wirelessly through an RF link to the implant. So just very briefly, I don't want to touch too much on the Orion study, but we've implanted 6 subjects overall. They all, of course, have acquired blindness, and this study is still ongoing. And I'll show you some examples of how we've worked with some of these subjects.
Starting now.
Again, using the nubbing. So this is just a sequence of phosphines, for example, that we've asked the subject to map out, and the subject uses a tactile fixation point and it tends to this location while we deliver electrical stimulation. And here the subject draws the location of each flash of light they see on the touchscreen, and that's clearly real time. You can see the timing. Sometimes the subjects would indicate the size and shape of each phosphine, except here in this task, we asked the subject just to indicate the location of the phosphine for each of the 5 subjects shown in 5 electrodes shown in slow sequence.
Starting now.
The image on the left shows where the Orion implant is arrayed, is positioned on the medial wall of the left occipital cortex in one of our subjects, and the image on the right shows the distribution of the phosphine locations obtained in the subject. You see a bit of an hourglass here, as one would expect due to the calcarine sulcus, and you can see that these electrodes span an area ranging from about 5 degrees eccentricity near central vision out to about 30 degrees. So as with earlier devices, the Orion subjects can perform some simple visual tasks with the device using the standard Orion mode. And we now have much better controlled and quantified experiments to develop it. For example, the subjects can locate high contrast squares on a screen in front of them, just using the standard second sight proprietary algorithms. They can determine the direction of a moving bar, and they can determine the orientation of a high contrast grating. I'll show some video clips of this particular subject performing some of these tasks using the proprietary algorithms.
Correct. On.
So you can see he's scanning the screen using the device largely as a light detector.
Correct.
And there you can see the camera view and the array activation. And this is not using dynamic stimulation. So now with the device off,
It was higher and right.
he consistently performs no better than chance. We do some testing with real world objects, and this is all part of the early feasibility study that Second Sight is leading. This subject's been blind, by the way, for over 20 years, no light perception whatsoever. Here he attempts to navigate the hallways near our laboratory with our research nurse standing close by. He points out the window.
Definitely see the sun.
That's a favorite of mine where he says, I definitely see the sun. I'm sorry about that. I'll go back to that point there. And you can see, of course, from his shadow as he points to the sun that he is quite correct. And, you know, on the human side of this, that was a rather emotional moment for the subject and for us, frankly. This slide here shows the performance for all the Orion subjects in the square localization task. You know, they're clearly much better than chance. The blue squares show with the system off and the black diamonds with the system on. And they're better at discriminating motion than they do at a chance level with the device off. But I think it's fair to say that a lot of this could easily be accounted for by just simply using the device as a light detector and scanning the screen using head movements to just perceive the areas where the device is activated by light. And we believe, and I think everyone would agree, that to really advance the field, we need to produce not just light detection, because that's far less useful and far less daunting a challenge. We want to produce some form recognition or pattern discrimination. So here's an example of one of our Orion subjects in Houston. And here we're using dynamic stimulation, the same strategy I showed you before. And in this subject, we use 4 different patterns that the subject would use to identify and discriminate dynamic stimulation patterns and correlate them with graphemes. In this case, each pattern contains stimulation of 5 electrodes, and we have some limits with our hardware and the software for the Orion as to how precisely and how widely we can deliver the stimulation. The image at the left shows where the electrodes are located in the brain, and the movie will show the first 3 trials of a 4 alternative discrimination task in which the subject was asked to draw the letter he perceived as he perceived it and to name it.
Starting now.
This subject also has no light perception.
Next one. Starting now.
You.
Next one. Starting now.
Yeah.
So here are the drawings he made in response to 10 different trials of each of the 4 patterns used in this discrimination task. And in this run of 40 trials, he got 37 out of 40 correct, well above chance. And he really only had about 3 to 5 trials of training in each pattern before this test. The only mistakes he made in the 3 trials he got wrong is where the actual answer was W, he reported U in terms of his verbal reports. Presumably, he just missed the upward sweep in the middle of the stimulation pattern. The actual answer was W, but he reported U. So this gives you a little look into what we were able to achieve with dynamic current steering, where we could not achieve anything close to this using these number of electrodes with static stimulation. So what are the problems with existing visual cortical prosthetics? I talked about our approaches to improving the stimulation pattern or software, but it's important also to think about the quality of the hardware. And we only had 60 electrodes in the Orion device. There are about 100 in a BlackRock array, and there are lots of biocompatibility problems with anything with stimulating electrodes. And of course, the other limitation that I talked about is this software assumption, the assumption that phosphines are equal to pixels. And I don't think this audience, especially Daniel, is convinced that phosphines are equivalent to pixels, but that's really been the prevailing assumption in the field. And I think it is valuable. I think if perhaps our most important contribution is that we are trying to break away from that assumption, because we don't believe that is going to be fruitful. We believe that will be hopeless if we just assume that they're just phosphines are pixels. And so we're part of a consortium that plans to investigate the use of new high density surface electrode arrays. And what we hope to use is use advanced hardware together with enhanced, what we call software, or enhanced stimulation paradigms, and to test new stimulation paradigms. So let me talk a little bit about that. So the novel hardware that we hope to use is called the Bioelectric Interface to Sensory Cortex, or BISC. And features of this device includes thousands of small high impedance service electrodes, integration of stimulation recording electrodes with an underlying complementary metal oxide semiconductor or CMOS electronics platform, integrated wireless powering and telemetry, a small form factor that allows implantation under the skull with no wires or connections. And the chip is rendered flexible through extreme thinning, which allows it to conform to the peel surface of the brain. Because the electrodes sit on the cortical surface rather than penetrating into it, surgical implantation is simple. And I've already implanted one of these with Andreas Talias at Baylor, a prototype, I'm sorry, in a monkey. It's not functional, but just to test the materials compatibility and flexibility and the like. And we've implanted and explanted in a single monkey, not having stimulated or recorded with the device, but the basic biocompatibility over several months is quite acceptable, and the ability to conform to the brain seems promising as well. And this next image will give you a sense of the size of the electrodes and how flexible it is. And you can see the number of electrodes magnified relative to the human hand. And here it's wrapped around a pen to show its flexibility. Obviously, other sensors are emphasizing modern or advanced versions of penetrating electrodes, which offers huge and different advantages. But from a safety perspective and an early feasibility testing perspective, there are some, and perhaps a long-term viability perspective, there are some real advantages to working on the cortical surface. And this is a little cartoon showing how we hope to advance from a BISC-1 module, which has 64,000 electrodes, up to BISC-2, that's the ambitious goal of ambitiously 1 million electrodes. And this work, of course, is not being done by me, but it's being done by Ken Shepard and a team of engineers. So in the second generation of this device that Ken's working on, we hope to have 1 million electrodes and hope to wrap this array around the occipital lobe and the occipital cortex to get maximal coverage of the early visual areas with V1 in red, V2 and V3 in blue and green respectively. Why do we need to have so many electrodes? Well, we want to cover a large area of visual cortex to give the subject a large field of view, and we want to have a high density representation in each part of the visual fields. So to conclude, there's a new generation of advances in visual cortical prosthetics that are emerging. We're not the only center. There are other centers working on this. And so far, the Orion visual cortical prosthetic has been demonstrated to be safe and to improve performance in multiple simple visually guided behaviors. We believe that understanding the science of brain stimulation and visual perception is absolutely critical to the success of these visual cortical prosthetics. But phosphines are not pixels, and dynamic stimulation we've shown in a very admittedly crude way, but I think powerful way. This novel approach to stimulation can be used to increase the amount of visual information that's delivered to subjects and allows them, in our experience at least, for the first time, to have some version of a crude percept that is coherent and matches a visual form. I want to, of course, acknowledge the large number of people who've been involved in this work, including Bill Bosking and Denise Oswald from my lab, and Ping Sung from my lab, Mike Beauchamp, my long-term research partner, as well as Brett Foster, Samir Sheth, Andreas Talias, who's highly involved at the BIS project, which is led by Ken Shepard, Nader Paratian, Soresh Nidakadad, and Robert Greenberg and Jesse Dorn, all from Second Sight. I want to acknowledge, of course, our EMU clinical partners and staff and our clinical trials office. And perhaps most notably, I want to really acknowledge the important efforts of our patients who really have been wonderful, frankly, collaborators in this effort, especially these Orion subjects who have volunteered their time going beyond the basic Orion clinical feasibility trial and allowing us to do some of this dynamic stimulation to test whether we can produce a more robust visual experience. And that, of course, is our goal, is someday to use a larger, higher resolution electroarray with strategic dynamic stimulation paradigms to produce perhaps contours of visual objects in various modes and create coherent percepts of visual forms. So thanks everyone for their attention, and I'm happy to take some questions at this point.
Thank you very much, Daniel. It's a remarkable hack into the visual system, and it's amazing that that instead of stimulating the pattern and not eliciting anything coherent, you just scan the same pattern sequentially, and you easily elicit a percept that very closely resembles that. So that looks like a pretty universal pattern, because you have shown that it works with different sizes of electrodes, which means that penetration depth and stimulation of different layers of the visual cortex is working with this modality. So that's very interesting, that you can stimulate to different depths and still this motion kind of is perceived as a proper shape, regardless how deep it goes. That's interesting. But I'm curious. You said that well, the scaling effort is now to 1 million electrodes. But if it only works sequentially, and so you need to avoid simultaneous activation, how many electrodes you can activate sequentially within a frame of the video? If you have 30 milliseconds, let's say, you may activate maybe 30 electrodes if you develop for
True.
a second. So
And I'm not saying that we're ready to give up on the concept of testing simultaneous stimulation of hundreds of electrodes at the same time, as many of our research partners are still quite hopeful that that will be an effective pattern. I would just say, speak from my own experience, attempting this on a lower scale, I'm skeptical that it will work, and I think we will have to use a dynamic pattern. I still think it's useful to have very broad coverage. You only get to implant the patients once, so you can't go back and move your electrode array afterwards. I think we want to test both paradigms and combine both in a useful fashion. But my early experience certainly suggests that simply turning on all the electrodes all at the same time to conform to a shape and assuming that they'll be perceived as combinatorial elements and building blocks, I think it's unlikely to be effective.
right. So if anybody wants to ask a question, you can raise your hand and unmute yourself. So to keep it in order, let's raise hands, and maybe we can unmute you just in order of okay, I see 3 already here. Just a sec. Karina, can you see the hands reason? Okay, I see here. Okay. So Bill, I see you are on top, so please go ahead.
Daniel, listening to this reminds me of psychophysics, motion psychophysics of 20 and 30 years ago, where we were all agog about long-range apparent motion. And, you know, we have these arrays of literally pixels on a screen, and when we alternate them back and forth at a particular frequency, you physically, I mean, you perceptually fill in the gap between those two, and you have a strong perception of, you know, of continuous motion of one dot to the other dot. And I'm wondering,
That's exactly what we were hoping to achieve, of course, is to fill in the gaps, as you say, and to create a unified form. I'm interested in your thoughts on this as an expert in motion, of course, visual motion. Would you have predicted this result, which I can only report on what we empirically observed, and I believe it was done with rigor. What are your thoughts about how this relates to your own notion of visual motion and form vision?
Yeah, I did not and would not have predicted it, but it, you know, everything makes some kind of sense in hindsight, right? Maybe we shouldn't have been surprised this happens, but the fact is I am surprised, and pleasantly surprised. I think it's impressive. And I wonder what the subject's reports are. Do they really report seeing something smoothly tracing out this letter?
So they report it's variable what they say, but one word they've used is skywriting is a term. I think they definitely, it's not, there's a dynamic element to the percept without a question, but it requires almost no training for them to translate these patterns into letters. So I think that speaks something for the perceptual quality of this. And, you know, with these Orion subjects, we're actually on hold now thanks to COVID, but we're hoping to enhance our psychophysical tasks and get to the root of their percepts in a more sophisticated fashion. But there's no question, I think a key point to your question is, this has not been done with exhaustive training. These are lazy experiments. We do them quickly, and the subjects catch on extremely quickly.
Yeah, so the phenomenon of long-range apparent motion is quite dependent on the timing between the two pixels that are flashed on and off of the video screen.
Well, we've used a range of stimulation timings, and we've had very similar results across different timings. We haven't looked at it rigorously in terms of systematically, but it has not been particular for one precise band of timing for us.
I see
really interesting. Thank you.
Hand is Charles. Charles, go ahead. David, if you want to, please raise your hand because I have a list here electronically. So can you raise it electronically?
I saw someone asked about color vision quickly. We'll be thrilled to get forms in black and white. There's no attempt at color vision in what we're doing. And I really think as a clinician, when you talk to patients, the key leap that we need to make in order to make this useful, there are two things we want to achieve. And one is object recognition in some form and the ability to navigate independently. And both those tasks really depend on having at least some rudimentary form vision. And that's what we're really aiming for. And that's the leap we're trying to achieve, that with dynamic stimulation, that had, to my interpretation of the literature, has not yet been achieved. If we can get a reasonable level of even crude form vision, I think we will really help patients. Yeah. Color is light years away from where we are.
Right. So next is Charles, and then David, and then Krishna Shenoy. Yeah, Charles, go ahead.
Yeah, thank you, Professor Yoshor. I'm a student in Daniel Palanker's lab. So I'm curious, how do you plan to extend the paradigm of dynamic current steering to more complicated real-world patterns? For example, human face, and you can't necessarily trace it along with just one trail.
So, you know, we've been talking with Jim DiCarlo about this, and what we kind of want to do is steal from the computer vision world and develop basically rapid contour and segmentation algorithms that allow the, you know, natural visual objects to be transformed into contours that can readily be traced out on the brain. And I think that also goes back to the idea, why do we want many, many thousands of electrodes? Because in the visual world, we won't be able to really reasonably assume that we're going to have a small number of graphemes that we're going to test. We need to be able to rapidly draw these contours and capture it. It'd be a screen capture, segmentation, a contour, and then a delivery of stimulation based on that contour. That's what we're hoping. We have no experience with that. Let me make that clear. We've only tested this using a handful of electrodes, and there's a lot of work that needs to be done. But that is the paradigm that we would like to test. And at the same time, we won't give up on the notion of testing concurrently stimulating many thousands of electrodes at the same time. But the other problem with stimulating thousands of electrodes at the same time to produce a useful images, then you run into the clinical problem of potentially producing a seizure. So the idea of using information segmented across time has another attractive advantage from that perspective too. Obviously, we have a very long way to go. This is just proof of a concept at this point.
David?
Daniel, great to see you again. Great work. I had a question about the gentleman who had been blind for 20 years at BCM that you were showing. I would have thought, depending on his age, that after 20 years of blindness, much of his visual cortex maybe was would be taken over by touch and hearing and so on. So I'm curious if he reported anything other than seeing.
So we were interested in that too, and we actually did some functional MRI testing using other sensory modalities. Mike Beauchamp did that. And we actually warned the subjects. We were a little concerned about this because, you know, again, the conventional thinking is, oh, you're working on a part of the brain that's doing nothing. And as you well know, that brain could be involved in language, for example. So we didn't see any robust activation in the limited screening we did with an fMRI. And that was, of course, before implantation. And we tested on a crude level whether there was any adverse effects to his language function, for example. And the Orion device has only been implanted on the left side. For some reason, when they designed it, you know, they're only left side right now. And I personally would have preferred the right side as a doctor looking to protect my patients. But we have not seen that, but it's certainly something we did consider, absolutely. We have not seen it.
Okay, Krishna, yeah, you should unmute yourself. Okay, yeah, go ahead, please.
Yeah, hi, Daniel. Wonderful talk, extremely exciting. I think your recent paper really lit up the field in a wonderful way. The question I have is regarding scaling, and this has come up in some ways. So this march towards higher quality vision and more useful vision would require scale up on the technological side, electronic side, which you covered beautifully. And I think one can envision that. But a strategic decision out of necessity, I think, is what you were describing in terms of using dynamic stimulation, which would seem to fundamentally limit what is otherwise this extremely high bandwidth of information coming into our brains by vision. So do you have thoughts on if you wanted to do fuller field, like parallel write-in, these types of things? I know this has come up in other questions, but just sort of fusing it together, is there a theoretical maximum that you've been able to calculate in terms of
within the constraints
by employing time and dynamical write-in, it seems to cap the bandwidth much lower than what vision typically is. And this is something that we on the motor output side are always limited by, because we're used to controlling one effector, and that sets fundamental limits on how much information you can get out, per unit time.
No, there's no question there's limits to the bandwidth. And I think, by using time, we actually enhance our bandwidth, of course, and how much information we can deliver. But there are a lot of constraints here. And again, people have to realize that producing seizures is a real issue here too. We can't stimulate endlessly, both from a functional viewpoint as well as a clinical viewpoint. So I think the idea that we're gonna come anywhere close to matching natural vision, of course, we're like
No. Yeah, so I'm not worried about natural, but so just one quick follow-up.
Sure.
So understood that there's a certain maximum, let's say, current that you can send in before triggering a seizure. So that's a clinical limit that brings down what you, okay. And then it seems to me like if you use time, you're not maximizing your bandwidth, you're actually limiting your bandwidth, because it takes, a second to write in a certain number of bits.
No, that is a limitation, but the problem is when we didn't use time, we got nothing. I mean.
No, I totally understand. So what I'm after is,
have we explored
calculation, say useful vision of a certain quality can be achieved having made the decision to use dynamic write-in? I think that would be extremely compelling.
I think that's a good point. That's something I really need to give some more thought to. It is a good point. Absolutely.
I think that'd be extremely compelling, because then you could say, listen, even though we're using time, we're still able to far exceed what people would have otherwise necessarily, maybe thought. Yeah.
Great.
Thank you. So the next question by.
Let me just grab my plug. Hold on 1 second.
Okay. Oh,
go ahead. I didn't want to have a power outage here. Sorry about that.
That was that me?
Yeah, please go ahead.
Okay, that was a really great talk. Thank you so much. I had a question. I was wondering if any of the patients that you've tested were congenitally blind, and if the sort of level of the training for these patients is that any different than patients that have.
Oh no, we purposely did not test any patients who are congenitally blind, and the assumption is that visual cortex is insufficiently developed to make this a useful strategy. So no, all the patients that we test are either sighted patients, of course, in the epilepsy monitoring unit and for the visual prosthetics. And the idea of using a visual cortical prosthetic pretty uniformly, we're going to restrict ourselves to subjects that theoretically have a well-developed visual maps. And as David pointed out, in fact, the concern is that even those patients may have repurposed their visual cortex, but certainly we're not going to try congenital blindness patients for this point.
So the next is Roxana and then Kalanit.
Hi, very interesting work. Yeah, thank you so much. I am curious to know how fast the cortex adapts to the stimulation like Orion stimulation. So you mentioned around 30 trials participants performed the word task, like to understand which letter you're presenting. Was it done in one session or multiple sessions?
No, we did those in one session. One session. We did not have, you're worried about adaptation, that the patients would cease to respond. We did not experience that. No, these are in single sessions. It's a theoretical concern, of course, but we did not experience that.
Kalanit, you should unmute yourself.
Okay, hi.
You're still muted, Kalanit.
Muted. Hi, can you hear me?
Yes.
Sure.
Okay, good. So I was really impressed by how intuitive it was for these patients to draw the letter in the video. That was really very impressive. And the contrast between the static and the dynamic in that blind was very striking. And I guess, well, going back to, it says that I'm muted by the, okay. Can you?
We hear you. The problem is we're hearing other people too. So
okay, I got the sign that I was muted by the host, so I just want to make sure that I'm not talking to myself. I guess I'm going back a little bit to Bill's question. It's like, so what's the magic with the dynamic stimulation? Why is the dynamic stimulation producing much more robust percept? And even if you don't know the answer right now, what would be your plan of attack to be to understanding this?
That's a great question and something I've really thought about. And that's in fact why I tried to pin that on Bill while I had him in the room. I told you the clinical motivation is when you're a first-year medical student, you're taught that the way you test somatosensation very quickly and very robustly is you draw letters on a patient's hand. And if you try to represent that letter with individual spots all at the same time, you can't detect the letter Z. But if you draw like this, it's incredibly intuitive. What is the reason this works in visual cortex? I mean, I think on a visceral level, we're hoping that we're creating some coherence by linking the spatial form across time. But I think you're also dealing with questions of inhibition, for example. Every time you stimulate the brain unnaturally, you're not just stimulating the pattern that would be produced with retinal activation, you're stimulating inhibitory interneurons and gosh knows what. And I think that this may help overcome that adverse input that would normally interfere with the percept. But I'm not entirely sure of why it actually works and what the theoretical basis for it is. It's something that we are interested in pursuing.
Thank you.
I have a follow-up question about the scalability that we discussed. The minimum duration of the stimulus that can be perceived and the minimum interval between two stimuli that perceived separately as a sequence in the scanning, what approximately are they?
So we're doing that now. We actually haven't done that yet. We really couldn't do that in the epilepsy subjects because we have such a limited amount of time to work with them. But now, and we were doing this until COVID put us on a hiatus, but we want to really define stimulating rapidly in succession. You saw some of the experiments that we started to work on when we do 5 at a time. We want to see sort of like a flicker fusion experiment in visual cortex. We don't have that data yet, but that's absolutely something that we're interested in defining in a way that I don't think, well, you certainly could do it in monkeys too, but I don't think it has been done in this sort of motivation.
Thank you. Yeah, Bill has another question. Bill, please unmute yourself.
Yeah, Dan, I definitely have ideas for experiments which you and I could maybe chat about a bit.
Let's talk about them. I'd love to hear. Yeah.
But one quick question. You might have said this already. I'm not sure, but your patients were tracing out these letters with their hands. If they were not using their hands and simply giving you verbal reports, could they just as easily name the letter, or was it something about tracing out the hand that helped them name the letter?
The answer is they could. And we did tests where we had them not trace it. They're not as the video is not as interesting for display, but yes, we did, when we wanted to test them even faster, we would just have them say the letter in sequence. Absolutely.
And no problem.
No problem.
Cool.
Okay. Bill, let's
like
follow up offline, please.
looks like we exhausted all the questions. So
can I say one thing quickly before we close? I want to thank Brian Wandell in particular. When I was first doing experiments in human visual cortex with John Mansell, who I should also thank, of course, my mentor in this space. And I was really kind of focused on doing basic visual neuroscience experiments. And I think it was really at a visit I made to Stanford, I don't know, about a decade ago, where Brian really kind of pushed me to go back to the Dobell space, he says, that had been neglected for so many decades. And he said, someone like you who's trying to do these experiments, and I say trying with humility, trying to be rigorous and quantitative in how we approach these experiments, should try to revisit the approach to using visual cortex and to try to help patients. So I think it's always kind of, I like to tell the story kind of ironically that it takes my PhD scientist friend to tell me to actually try to be a help patients doctor. So I want to thank Brian for that motivation and inspiration, and it really did make a difference in me kind of visiting this space.
I think the most remarkable discovery, which is kind of unexpected, this sequential activation and current steering suddenly produced very coherent output. You couldn't predict that. Maybe somebody could, but nobody tried until now. And suddenly it's a world of difference. Instead of applying the same pattern simultaneously, we apply sequentially and get tremendously different results. So whose idea was
it? I guess I would really give the credit to Bill Bosking, my research associate. We talked about it, and we talked about this concept of tracing, and it was a discussion with Bill that really, I think Bill deserves the lion's share of the credit. And we talked about this concept of tracing objects. We were frustrated. I mean, the root of the conversation was we were frustrated that we were stimulating everything at the same time. And we have a paper coming out describing that raw data. It's not as pretty a story. And Bill has tried to develop a rigorous rules to predict the patient's perceptual result. But sometimes you stimulate 5 electrodes at the time and they see 3. Sometimes they see 1 blob. Sometimes they see 2 distinct blobs. There's competitive interactions. It was a terrible exercise in frustration. And the root of the discussion that led us in this direction was, how do we get them to see